Let's see. There you go. Well, good morning, everybody. I guess it's 9. So we'll wait just a few minutes and then get started.
Yeah, it's funny what you were mentioning, Steph, about people using your account, because there's on the list there are often certain people show up 3 or 4 times.
Right.
Yeah, that's funny. Very nice.
Well, that's a good way to do the contact tracing if somebody misbehaves.
That's right. Oh, boy. This is the life. It's the weird life. All right, we'll wait 1 more minute and then we'll get started. Thanks for tuning in, everybody. So, as always, really great to see everyone. I'm going to give a shout out to Navish. Good morning, Navish. It's always funny to go through the list of all the peeps.
Mm-hmm.
Alums, if you want to send Aaron and me mails, we'll be very happy to hear from you. It's just kind of a pity we don't all get to chat. It's the real Zashen. Greetings. All right, I'm going to get started. So welcome to this morning's PBOC lecture. It's once again a really great pleasure to introduce today's speaker, who is Professor Steph Weber from the Department of Biology at McGill University. As usual, I'll start out with some of the facts, which end up being less interesting than the parts that come after. Steph did an undergrad degree at Duke University, where she majored in both biology and chemistry, and not surprisingly was a superstar. And from there went to Stanford to do her PhD. And that's when I got to know her, because she did her PhD with my beloved friend and collaborator, Julie Theriault, who's on the call with us right now. And then after that, she went to Princeton, where she was a postdoc in the laboratory of Cliff Brangwyn, and did some beautiful work, which I'll comment on again in a minute. And then from there, went to McGill University in Montreal. And the thing I wanted to say is that, by the time you get to be my age, you have I'm going to call it the privilege of writing letters for many people. I've written letters for many of you that are on this call. And I'm going to just say, for me personally, I generally don't write letters for people that I'm not highly supportive of. And one of my favorite things to say is that this or that person touched me so much that we ended up writing about their work in our book. And Steph doesn't know this, but the 5 of us, Julie, Janne, Erna, and Christina, and I, we often refer to the 700-degree cytoplasm, and it's always with reference to Steph's grad work, which she did with Julie and Andy Spakowitz. And I love to be able to say, that's textbook material, but it goes farther than that, because then her postdoc work, it's the same deal. So again, she doesn't know this yet, but we actually have multiple figures in the new edition of our book, which are based on her postdoc work. And in my opinion, I'll embarrass her a little, like I did probably Amy Gladfelter yesterday, but she's really got the Midas touch. And she comes to problems and does really beautiful work that leaves me as a theorist wanting to think about it. And that's going to be true for today's story, because now she's going to tell us about bacterial condensates having to do with RNA polymerase, and something that my group members and I are deeply interested in. Steph has a really great connection to MBL. She's going to have to remind me if I got this right, but she came as a TA with Julie and Natalie Dye also. And if I'm not mistaken, Steph, you'll have to comment on this, but I'm almost sure that you and Natalie stayed at my house on Devil's Lane. And I know that the 3 of you, Julie and Natalie, found the whole experience pretty provocative and intense, which we all know it is. And it was great to meet you back then. But as I told you in a recent email, it's just been so fun to reconnect. I loved my visit to see you last year at McGill, and I tip my hat to you. I mean, really, for the work that you've done. That I'm going to tell it this way. You're not in our book because you're a Julie student. You're in our book because you did such cool stuff. Anyway, so I'm super excited about your talk, and I love the title, No Membrane, No Problem. It's sufficiently chill, funny, and cool. And now that I've embarrassed you, I'm going to great for you.
All right. Wow. Yeah. Thanks, Rob. I feel like I'm 700 degrees now. Yeah, thank you. So I'm thrilled to be part of this the seminar series. Wish we could have been there in real life. I don't think we stayed at your house. I was definitely in Swope or something like that. But yeah, Natalie and I would have actually said, everyone else in this seminar series has said how much MBL meant to them and how much they loved their experience there. I actually hated my experience there. It was actually pretty miserable and intense is a very euphemistic word, but it certainly made me think and have an existential crisis about my PhD thesis. But all of the sort of ideas and ways that you and Hernan approach science have certainly stayed with me, even if I didn't learn those lessons at MBL. So Rob mentioned, why can I not advance? Okay, so Rob mentioned that he visited McGill last year. So we're starting a molecular and cellular biophysics center, largely inspired by the PBSC book and what he's done at MBL and Julie and Hernan and everyone. And I love this picture because Rob is, waxing eloquently, and I look like a idiot fangirl geeking out over him, and then everyone else is just like, what is he saying? Anyway, it was a real thrill to have you visit, and I'm thrilled. At the time, we didn't really have much of a story to tell you yet, but we do now, and so I'm thrilled to share that. So I'm gonna start way back with an analogy. So when Robert Hooke first looked through a microscope and saw cork cells, they reminded him of the rooms in a monastery. And so the word cells actually is Latin for small room. And I'm going to use this today because I'm going to use a room, and welcome to my bedroom in COVID-19 era, to describe entropy and sort of why I think organization is an interesting question in biology. So when I was a kid, I used to read the Berenstein Bears books, or my parents read it to me. And there's this one about the messy room. And what I want to just paint a picture for you is a child's bedroom can be a mess, right? And so unless you put work in and exert energy to maintain order, things will just go to chaos, and you won't be able to find things, and you'll trip over things. And so like a bedroom, cells function best when they're organized. And so here, again, the toys are strewn all around, so you could trip. If there's old dishes, that could be some sort of health hazard if something starts growing. But if you clean up the cell and you put your toys away in the toy box and you make your bed, then everything is in its proper place, and there are no safety hazards, and you can find things quickly and easily, and things function. And so the same thing applies to a cell. So cells are organized into functional compartments. So just like an organism contains organs, and each organ has a specific function, and its structure matches its function, the cell is subdivided into organelles, or little organs. And so this is a classic textbook image of a eukaryotic cell, where you have a big nucleus in the middle that's containing the genome, and then a whole bunch of other membrane-bound organelles distributed throughout the cytoplasm. And so what I want to emphasize is that most of you probably, when you learned about biology in a textbook, learned about membrane-bound organelles. And so in this old EM, you can see the membranes sort of enclose all of these compartments in the cell, and membranes are electron dense, so you can really see them. And they're well preserved by these ultrastructural techniques. And so the function of this membrane is to impose a diffusion barrier between an organelle and the cytoplasm, such that each compartment can maintain a distinct biochemical composition and specialize those molecules for whatever its function is. So the DNA protects the genome, mitochondria make ATP, and the ER and the Golgi package and sort proteins. And all of these functions are made possible by membranes. And the assumption until actually it's almost a decade ago now, but was that the cytoplasm is just well mixed because there's no membranes, so molecules are free to diffuse around, and you won't have any sort of concentration gradients because it'll just entropy will win and the cell will be dirty and a messy bedroom. Until about 10 years ago, this was sort of the concept of intracellular organization, that membranes enclosed organelles, and that was the way that cells could specialize functions. But that only applies to eukaryotes. And this is one of the latest trees of life. And I just want to point out that eukaryotes and Hopi's charismatic mini fauna are this one teeny tiny twig of a branch, this green branch on this tree of life. And all the rest of these organisms are prokaryotes. And prokaryotes, at least so the textbook says, don't have any internal compartments. They have DNA that's not enclosed by a membrane, and they have ribosomes kind of floating through an empty sea of goop, cytoplasm. And so the question is, well, how do prokaryotes tidy up and organize and clean their rooms? And if they don't, how do they have specialized functions if they don't use membranes? And so this is the basic question that I want to pose today. And so getting away from cartoons and into actual images, this is an electron micrograph of an E. coli cell in contrast to the mammalian cell that I showed you earlier. And you can see the dark membrane on the outside where the plasma membrane is, and then there's not much structure on the inside. You can see a little bit of electron light nucleoid. The DNA is the white stuff here, and then each of these black spots is a ribosome. So there's a little bit of separation between the nucleoid and the cytoplasm that's filled with ribosomes, but there's certainly not a membrane, and there's no physical barrier. However, nevertheless, many molecules are still able to not be just one big homogeneous chaotic mess, but they localize into non-uniform patterns. So proteins like FTSC forms a ring at the mid cell. Chromosomal loci, which Rob alluded to earlier, I looked at in grad school, occupy specific locations in the cell. mRNAs also are tethered to their site of synthesis, and even lipids collect near the poles because of the curvature of the membrane. And so we can, with all this new fluorescence microscopy and super resolution imaging, we can see these patterns in bacterial cells, but we still don't know much about how this order arises. And so,
hey, Steph.
As I mentioned, they don't use membranes, so there must be some alternative mechanism.
Hey, Steph.
Yep.
Can you hear me?
Yeah.
There's a question related to this, kind of playful, but I think it's definitely an interesting one. If your room is 1 micrometer long, do you need to clean it? It can be easy to find your toys that diffuse around on a timescale of 1 second.
Yeah, no, so that's a great question, because I think a lot of the sort of hand waving assumptions before we were really asking these questions was, well, maybe they're not necessary. Maybe you don't need membrane-bound organelles because it's so small and diffusion is fast enough, and you can just have a well-mixed system. And so that's actually, so I'll come back to sort of the function of organization at the end of the talk. And so right now we're just trying to look at, is there order and how does the order come about? But the elephant in the room is, what is the function, what is the purpose, and is it necessary? And so, yeah, I think it's probably, for some things, it's probably less necessary in bacteria than eukaryotes. But I'll talk about a couple examples where it does become necessary. Yeah, I hope that answers that question. It's a good question. Okay, so I just mentioned that bacteria don't use membranes, and now I'm going to tell you about an example where they do. So one beautiful example of a bacterial organelle are these magnetosomes, which are help bacteria align in the magnetic field in the water column, and they are just invaginations of the inner membrane. And so these are magnetite crystals, and you have a row of them aligned with a actin homolog. So the membrane paradigm does exist in a few examples in bacteria. There are also storage granules that don't have a membrane around them, but are sort of aggregates of insoluble long chain polymers. And then there are the canonical bacterial microcompartments, which have these, like the carbosome, which have these beautiful icosahedral shapes, and the shell of these are protein based. And this also creates a semi-permeable barrier that allows Rubisco to, or that allows the cell to concentrate carbon dioxide so that Rubisco can fix carbon. Anyway, okay, so those are at least the mechanisms that have been described to form compartments in bacteria. But Amy Gladfelter mentioned yesterday that phase separation is another mechanism for organization in eukaryotic cells. And so here we can see germ granules in the C. elegans embryo. Cliff took this in his postdoc. Actually, this might have even been taken with Toll. And what you can see here is that P granule proteins are not well mixed in the cytoplasm, but instead condense into droplet-like things. The droplets don't have a membrane, and so they're still able to individual molecules are still able to exchange. And you'll also notice here that there is a asymmetry. So there's a partitioning of the P granules in the posterior end of the embryo. And this is due to a concentration gradient of other proteins that we can't see here. But this also shows that the dynamic rapid assembly and disassembly of the germ granules can respond to developmental cues rapidly, because they dissolve in the anterior and they condense in the posterior. And then in addition, the membraneless organelles can respond to external environmental cues. So here they've tossed in arsenic into the media. Cells don't like arsenic. So they form these stress granules, which you can see a fusion of these droplets here. And importantly, this is reversible. So when the arsenic is washed out, the cells or the droplets dissolve back into cytoplasm. And I think this is looping back again. So membraneless organelles and phase separation give cells a mechanism to rapidly respond to developmental or environmental conditions to form transient compartments. And so this is, again, just to drive home the point, you can have a well-mixed cytoplasm or nucleoplasm in this case, or a phase-separated nucleoplasm where you have two phases that are in equilibrium that have different compositions and without a membrane between them. And so Amy brought up a lava lamp yesterday. If you got, I don't actually like this analogy, to be honest, because a lava lamp is adding heat to the system. So this is already non-equilibrium, and that's a whole nother bag of worms that I'm happy to talk about later. But another analogy is that you can just think of mixtures of liquids. So alcohol and water mix, right? And so this is a homogeneous solution in your beer, but they can also, two liquids can phase separate, like oil and vinegar in your salad dressing, and you can have two different phases that are not separated by a physical boundary. Okay, so the question that I wanted to ask when I started my lab was, do bacteria use LLPS, liquid-liquid phase separation, for intracellular organization? And my rationale was that, okay, we know dozens of examples of liquid-liquid phase separation in eukaryotic cells, but kind of counter to the question that was just posed, rather than asking do bacteria need it? I was kind of going from the opposite end, right? Like if bacteria don't use membranes to organize, they have to use something. And so why not use phase separation? It seemed like that not only it was likely to exist, but second of all, it might be more functionally important in bacterial cells because they don't have membrane-bound organelles as an alternative. And one of the challenges in eukaryotic phase separation field has been measuring the function, partly because it's just hard to measure like with experimental tools that we have currently, but also because some of the functions seem to be like small tuning things, like a little bit here and a little bit there, because the membrane is what really does the bulk of the partitioning. And so my rationale was that in bacteria, maybe phase separation would be like the the big functional thing, and so maybe it would be an easier system to study the function of phase separation. Okay, so I started posing this question in like 2016, and then realized that when I was born, people were already thinking about this. So in 1984, there's an old paper where they were using refractometry to look at bacterial cells. And what you can see is that there is a difference of index of refraction in the nucleoid relative to the cytoplasm in E. coli. And so they use this as a way to show that the macromolecular density inside bacteria is not homogeneous, and there are already at least two compartments, right? There's a cytoplasm that has a lower index of refraction, and then there's the nucleoid where the DNA is that has a higher index, lower index of refraction, and that you already get phase separation between the DNA-rich nucleoid and the ribosome-rich cytoplasm. Okay, and so that's very cool. And there's a lot of people looking at sort of nucleoid organization and chromosome organization in bacteria. We wanted to look at a slightly different system. And the system that we chose as a candidate for phase separation was bacterial polymerase, so RNA polymerase, like Rob said, partly because Rob has done, and Hernan have done so much beautiful work on how transcription is regulated. And also this is sort of analogous to the nucleolus, which I worked on in my postdoc in eukaryotes. And so the basic observation that was made back in 2003 is that RNA polymerase in E. coli is roughly uniformly distributed throughout the nucleoid in slow-growing cells. But if in fast-growing cells, so in rich media, when nutrients are really abundant, then you get these clusters of RNA polymerase that concentrate into spots throughout the cell. And it wasn't at all clear how what caused the clustering and what the sensing mechanism is, right? Like why are they not clustered in minimal media, but they are in rich media? So this was a system that we chose to sort of explore. And I thought this could be a really rich place to do functional analysis after we characterize just the clustering itself, because there's this connection to nutrient availability and growth. Okay, so when I first started my lab, my first postdoc, Anne-Marie Ladessar, spearheaded and basically completely started this project from scratch because I didn't even have the strains, and she worked with a really talented undergrad, Graydon Tope. And so they did a very simple experiment. So previously people had just looked in log phase cells in minimal media or in rich media, but we wanted to look at the dynamics of assembly and disassembly. And so what Anne-Marie did was she took a saturated overnight culture, she diluted it into fresh medium, and then she watched the distribution of the polymerase over time in cells. And having done live cell microscopy in Julie's lab and in Cliff's lab for my postdoc, was convinced that we could watch this live. And so Anne-Marie, took her cells from the 37 shaking incubator, ran over to the microscope, and all of these, beautiful images that we had seen in the literature looked like this. even in fast growth rich media. And so it turned out that the clusters of the polymerase were so dynamic and so sensitive to the environment and the surrounding medium that they disappeared and disassembled before we could even look at them. So unfortunately, initially we ended up doing just a fixation time course. And so the first data set that I'm going to show you is taken from fixed samples. And so she sets back the culture and then takes a sample every 30 minutes, and then we measure the spatial distribution of how the polymerase changes. Okay, so in stationary phase, the polymerase is, as I said before, is roughly uniformly distributed throughout the nucleoid, and it has roughly, so this is a pixel intensity histogram. So about 10% of the pixels are dark, and about 10%, or maybe a little less, are light, and so you get roughly a flat distribution. But in log phase at 120 minutes, you can start to see bright spots and really dark background where there's no nucleoid, and then kind of like intermediate background in the nucleoid, absent the bright spots. And so this, you can see a shift in the histogram to the left towards lower intensity values, and that's because there's more dark background. And so these bright pixels correspond to the clusters, and this increase in dark pixels is the background. And so we can set just an arbitrary threshold at 50% and say, okay, the number or the fraction of pixels that is above this threshold is the sort of metric of clustering. And so to validate this metric, we use two controls. HOP-A is a HU protein. It's a nucleoid-associated protein that's supposed to bind to DNA relatively non-sequence specifically. And so here you can see a relatively uniform distribution. In contrast, LAC-I is going to bind to a specific sequence that we've inserted into the genome. And so you see 4 discrete spots, and then there's no LAC-I anywhere else. And so you get a lot of dark black pixels. And so here you get a low degree of large, I'm getting this backwards. Sorry, you get a small number of bright pixels and a large number of dim pixels, and so the clustered phenotype is a high number of dim pixels. So it's actually, the threshold is actually, we're counting the number of pixels below the threshold. Sorry about that. It'll hopefully become a little bit clearer in a second. So here, what we've done is we have the whole time series, so from 0 to 360 minutes, and log phase is in the intermediate sort of 120, 180 minutes. And what you can see is starting out, all of the cells have roughly 50-50% of pixels underneath this 50% threshold. And so that means it has roughly a uniform distribution. And as the cells enter log phase, you get a large population of the cells now have a really high degree of clustering, meaning they have a lot of black pixels because most of the polymerase is clustered into a few bright foci. And then as the cells re-enter the next stationary phase, they start to disperse again. And here the HOP-A that is a dispersed control is the light blue, and the LAC-I that is a clustered control is the dark blue up here.
Hey, Steph.
Yep.
One question is, does the spatial distribution of the genome also change depending on the media conditions? Yes,
and that's a really good question. It absolutely does. And so one of the reasons that we wanted to use HOP-A as a control is because HOP-A is also going to reflect the changes in the chromosome. So the chromosome, first of all, it under-reduplicates, so you get more copy, so you get just a higher copy number of DNA, and it also becomes more compact in the rich phase and log phase. But the HOP-A should reflect that. And so you do see a little bit of increased clustering in the HOP-A in the log phase relative to the stationary phase, but the clustering that we see from the polymerase is clearly well above that. But that's a good question.
Okay, and something also related to this, does the clustering of RNAP in rich media correlate with the size of the cell, which appears to be varying in time? So that's from Navish.
Yes, that is a fantastic question, and it's certainly correlated, but I don't know which direction the causal arrow is, and that's something that we're definitely going to I'll talk a little bit more about later, and we are definitely looking into, but I don't have a good answer for you yet.
And let me ask you a slightly technical question, though. I think it's relevant for this. So all of these are fixed snapshots, right? You're fixing the cells.
Yes.
Okay. And so is it so, like, how were you trying to image it with live imaging? I'm trying to think of the type of stuff that Tom Coleman did, for example, using turf, where he could get a lot of mileage.
Yeah. No. The problem was not that it can't be done. The problem was I didn't have the equipment to do it.
Okay.
So I didn't have an environmental chamber on my microscope. So we were imaging at room temperature. And so, this was taken on a confocal. So later in the talk, I'll show you live cell imaging on a turf microscope with a 37-degree incubator, and it's fine. Yeah, it was just a technical thing that we were a new lab and we didn't have the equipment yet. Okay,
so it's not that fast. It's still, you can still see.
Yeah, no, right. Yeah, it was just like within a couple minutes from going to the incubator to the microscope, they dissolve. But yeah, you could easily look at it if you, it's really the temperature more than the going from liquid to a pad, like a solid surface. It was just the temperature, 37 to room temp.
Got it, thanks.
Yeah, no, it's a good question. Okay, so going from stationary phase to log phase, you clearly see a change in the distribution. And I want to point out this kind of intermediate stage. So it could be just like, some continuous shift from dispersed to clustered. But what you see here is actually kind of a bimodal population where some of the cells are like a little bit late and delayed in exiting log phase, and they still are dispersed. And some of the cells have already clustered their polymerase. So there seems to be sort of a discrete transition in distribution of the polymerase. Okay, so as the question said, the cells grow large and fast in rich media. And so we also wanted to compare with minimal media. And so here you can see the cells grow, and there's some clustering, but the cell size is smaller, right? It doesn't grow as large. And the growth rate is also slower. And the degree of clustering is less in this minimal media compared to what we see in rich media. So we're really interested in comparing these subtle differences. Most of what I'm going to show you for the rest of the talk is just focusing on the rich media, just because the clusters are big and obvious and easier to experimentally interrogate. But there's a really interesting correlation here. So actually, before I move on, so here I'm going to plot the clustering, percent clustering as a function of doubling time. So in LB at 37, we get a high degree of clustering and a fast or short doubling time. And in M9, we get a lower amount of clustering and a slow growth rate, or a high long doubling time. And then we've done a couple other conditions in between. And so here we have in rich media, but at 30 degrees. And so you can see a decrease in the clustering and sort of an intermediate doubling time. And so these results are very consistent with previous single molecule imaging from other groups. So this is from Achilles Kaponitis, where he showed that in rich media, the cells are large. You get large clusters with lots of polymerases per cluster. And in minimal media, you get small cells with a few clusters, and the clusters are relatively small. And so Rob told me that I could wildly speculate, so I'm going to, and I'm going to fit a straight line between 3 points. And I realized that this is slightly ridiculous, but I was really intrigued that it was a straight line, because there are these things called bacterial growth laws, which are these empirical relationship between the ribosome content of bacterial cells and the growth rate. And so over a variety of different media conditions or antibiotic conditions, so like poison, toxins, you can manipulate the growth rate of the cell, and that changes the amount of RNA to protein ratio, which basically corresponds to the ribosome content. And so the hypothesis is that these clusters form around ribosomal RNA genes, which are the most highly transcribed genes, especially in rich growth media. And so maybe you have this, maybe this correlation between clustering and growth rate is sort of the molecular mechanism for this growth law, where in faster growth rates, you have more ribosomes because you have more polymerases around the RN genes. So that's, I have no evidence for that, but that was like a really intriguing observation for me. Okay, so the next thing we wanted to do was really ask what drives clustering of the polymerase. And so there's two really obvious hypotheses. So the bottom one, liquid-liquid phase separation, is how I set up this talk, and that's the one that I was most curious about and most hopeful to find. But there's a really more obvious null hypothesis that the first few times I was talking about this project to various people, they thought that I was clearly insane and crazy and stupid for thinking that LLPS might be responsible, because obviously it was DNA binding. And so now I'm gonna, per PBOC, kind of what are the states and weights and quick back of the envelope calculation, what, could DNA binding explain all the observations I told you so far? So, so if you take a cell and you lyse it and you spread it out on an EM grid, these are called Miller spreads, you get these beautiful, what are referred to as Christmas trees. And so what this is this line here is the DNA. And then the fuzzy lines coming off is RNA, and then the dark thing, the dark big blob at the center along this is the polymerase. And so what you're seeing is bumper cars of polymerases lined up on ribosomal RNA genes, and then you're seeing the progressive synthesis of the gene as you go from left to right. And so from these classic EMs, they calculated that there's one RNA polymerase every 85 base pairs. So they are like really, lined up train car by train car along the RNA genes. And so you could clearly just have high concentrations of polymerase at specific locations in the cell just because they're transcribing highly transcribed RNAs. So, to sort of figure out whether this was quantitatively consistent with the observations, we did just a quick back of the envelope calculation. So, just a little bit of background. E. coli has a circular chromosome, and there are 7 ribosomal RNA operons distributed throughout the chromosome. Most of them are near the origin, and this is thought to be that when the DNA starts replicating, you get multiple copies of this, right? So the cell, there are 7 copies in the genome, but cells actually probably have, 30 to 50 copies of this as the chromosome endoreduplicates before the cell divides. And so each one of these is a gene, and so you can have, a polymerase every 85 base pairs on this gene, on this operon. And then this is a little bit controversial, but there's some evidence that these RN operons co-localize inside the cells. And so at slow growth, nutrient poor media, you have one operon or one cluster of operons, and as the cell as growth rate accelerates and there's more media, the cells grow faster and you have more RNs. But RND, this guy, and RNB, this guy, co-localize in the same location, even though they're in distant spaces in genomic space. And so given this, we took the length of the operon, 5.5 kilobases, the density of polymerase on these operons and the number of RNA operons, and got a very conservative lower estimate of 453 RNA polymerases bound. And again, based on the Capanitas single molecule work, they had counted sort of in the hundreds of molecules per cluster. And so this is comparable given all of the assumptions that we made, and says that, okay, DNA binding is clearly sufficient to give you clustering. And so why you don't need to invoke LLPS.
Can I make a comment though? Or
yes,
ask
please.
A question?
Yep,
yeah,
yep. Because
the RNAP copy number runs, say, between 3,000 and 12,000. And so I'm going to claim that even though that cluster, like what you just said, I'm a little bit rejecting that null hypothesis because the background is still going to be, I would think, very high in that case. Although, you'll still have to answer that same question if it's phase separation too. But anyway,
yeah,
I'm curious what you have to say about the overall copy number.
No, that's a great point. And the copy number of the polymerase changes from stationary phase to log phase, and it's different in minimal media and rich media. And that's an absolutely excellent question. And I haven't addressed it, and we can talk about it at the end. Yeah, no, for sure.
Okay, thanks.
Okay, so maybe DNA binding can or cannot, whether you're convinced by that argument. A lot of grant reviewers certainly were convinced by that argument and thought that the LLPS proposal was bunk. But anyway, rather than just using that, we really wanted to do some sort of quantitative experiment where we could rule out one or the other experiment or model, rather than just using this estimate to say, oh, okay, it must be DNA binding. And so this leads us to the question of how do we test LLPS in cells? And so not only in bacteria, but even in eukaryotic cells, this is not a trivial thing. So thus far, the field has kind of come up with a couple of criteria that are required to conclude that LLPS is happening in cells. And so one is that you have droplets that fuse. So like if you have two liquid droplets, they will fuse and round up, and the volume will be conserved. Another one is spherical shape. And so if it is a liquid, then you should have, there should be a penalty to having interface. And so the system wants to reduce the interfacial tension and reduce the surface area between the droplet and the surrounding medium. And then the molecules should be mobile. So they should be able to move inside the droplet itself, and they should be able to exchange from the outside in and from the inside out. And so the way that these have been done is to look at droplet fusion. People have taken time-lapse movies of live cells. And so you can see here in mammalian cells, stress granules, multiple droplets nucleate, and then these two fuse and round up to form a spherical droplet. The shape can be calculated by calculating the aspect ratio of droplets from images, and they can be still images or movies. And so here is a distribution of nucleoli in Xenopus oocytes, and they have a peak that is highly spherical. And then the most common tool to use to measure molecular mobility has been FRAP, or fluorescence recovery after photobleaching. So here we have a P granule, and what they've done what's called a half bleach. So they take a high intensity laser and they bleach the fluorescence in part of the granule, and then they look to see if you get recovery, and you can actually get two types of recovery, right? You can get recovery from this other side of the granule, so you can get internal rearrangements of molecules, or you could get recovery from the outside and have sort of exchange. But this is the one of the sort of classic techniques that are used to show liquid phase separation. We can have lots of debates after if this is a good tool, and there's a lot of controversy, but this is sort of the current standard that the field uses. Okay, and so
hey, Steph,
yeah,
I got one question. It's related to one of the to a question by Shri Yervisvas. Well, she's asking, the speculation regarding single cell growth rates change via media, nutrient, or antibiotic exposure, but not by changes in growth temperature. So, so she's asking whether temperature is playing a role, and I wanted to use the chance also to ask you about your opinion about temperature as a tuning variable here. Bacteria can, I guess, grow at a wider range of temperatures that most mammalian systems that you probably, or most eukaryotic systems that you work with. So, but I know that it changes a bunch of other things in the cell. So I was curious whether you could comment a little bit on temperature as a tuning knob.
Yeah, so I'm not sure I totally got the full question, but so cell size and growth rate absolutely changes with temperature as well as nutrient or growth phase. And so we see higher degrees of clustering at higher temperatures than lower temperatures. If you grow them at cold enough temperatures, that they still grow, but they're super slow, and you don't see any clustering. We've also are starting to do sort of heat shocks and sort of see how high you can go, and then you have this whole stress response. But yes, temperature is absolutely an awesome control variable that we've only started playing around with. But I think what's nice about temperature is you can change it very acutely, and so nutrients, there's not only a difference in cell size, but there's also a difference in composition, right? Like the copy number of various proteins is very different. But you can take a cell that's been growing at 30 degrees and bring it up to 37, and in the short term, you won't have the composition will be the same, and so you can look at how temperature affects either phase separation or DNA binding or whatever. Temperature is a fantastic parameter. We haven't really we've only started to scratch the surface, but that's something that we're totally interested in, and we now have the incubator on the microscope to do that. So yeah.
And so Shri sent a clarifying thing. So what she was asking is whether these growth laws that you were positing that the data that you had looked like, okay, whether temperature is also playing a role here in this growth laws, I guess.
So now that you say that like point blank, I can't recall if people have done those growth laws at different temperatures, but I would be shocked if it didn't hold. It would just be shifted, along that diagonal. But I can't off the top of my head recall.
Mm-hmm. Okay.
But the size certainly, everything else changes with temperature. So I would bet money on it, but I can't recall the experiment right now.
Got it. Thanks.
Yep. Okay. So all of these methods to look for phase separation in cells have been developed on eukaryotic systems, which are huge. So this is the C. elegans embryo, which is about 50 microns across. The P granules inside the C. elegans embryo are several microns across. So this one is 4 microns. And so I just want to, per PBOC, tradition, bring back scale to you. So this is an E. coli cell. It would fit inside a P granule. And so all of the tools that have been developed to interrogate phase separation in eukaryotic systems are really not well suited for E. coli because they're so freaking small. And so trying to see droplet fusion or to measure aspect ratios for structures that are below the diffraction limit is really challenging, right? So I'm going to come back to molecular mobility. But one thing that we used in addition was this chemical solvent. So there's a whole nother can of worms with hexane diol, but I'm going to bring it up. So hexane diol is it's just a chemical, right? And it's polar enough that it can enter cells, but it is hydrophobic enough that it disrupts hydrophobic interactions. And so it has been used because it can dissolve certain types of membraneless organelles that people had by other methods shown were phase separated, but it is by no means necessary or sufficient evidence for LLPS. So just as an example, In yeast, p-bodies are thought to be phase-separated condensates. So if you mock treat yeast, then the p-body is stable. If you add hexanediol, the p-body disperses. And if you wash out the hexanediol, then the p-body reforms. However, if you look at prions, a protein that undergoes a conformational change to form amyloids that aggregate, they are resistant to hexanediol. So hexanediol has been used to distinguish between liquids and solids, if you will. And so we just wanted to see whether the RNAP clusters that we found are sensitive to hexanediol. And so here we have the mock treated, so you can see the clusters. When we add hexanediol, the clusters disperse, and when we wash it out, they come back. So that was encouraging. It's also dose dependent. So that also suggests that it's acting as sort of a solvent. And so if you have a lot of hexanediol, then it sort of disperses as if you had no clusters at all. Okay, but the real sort of control for us was to see, this is the first time people had used hexanediol in bacteria. Who knows, maybe it disrupts everything. This is an acute treatment. They certainly do not grow well in hexanediol. So we overexpressed the polymerase subunit that we've been looking at, and it forms inclusion bodies. So it forms these aggregates at the poles of the cell and at the mid cell that are thought to be amyloid-like. And then we also used LAC-I, which is a DNA binding protein, sequence specific, to see if hexanediol would disrupt protein DNA binding. Sorry for the build. And so we don't see any change in LAC-I binding or in the overexpressed aggregates when we add hexanediol. So there's at least something physically different about the clusters relative to protein binding to DNA or protein aggregating into amyloid-like structures. Okay, so then the next thing that we really wanted to do was look at molecular mobility. And the hypothesis here was that if it was the DNA binding hypothesis, then RNAP should have the same mobility as a DNA locus, right? But if it was in a phase-separated condensate, then maybe it would diffuse more quickly. And so to do this, we used single molecule tracking. So the basic idea here is that we use a fluorophore that can be photoactivated or photoconverted so that we can look at one molecule at a time. And then we can fit a Gaussian to each spot and very precisely measure the position of the molecule. And we can do this over time. And so if the molecules are well spaced out, then we can detect and find the position of each molecule. And so then we can do this iteratively, right? So we have a cell that has, as Rob mentioned, thousands of copies of RNA polymerase. We activate it, and then we're only looking at one of them. We can watch it diffuse around the cell, and then we can collect iteratively tracks of many, many molecules over time in the cell. Okay, so this is what my PhD student, Bajot Parmat, did. And so he activated continuously and then tracked individual molecules. And then at the end, he projected all of those time series onto a single frame and looked to see where the densities of these tracks were distributed throughout the cell. So this is a very early, poor image, but just to give you an idea. So the green is where all of the molecules are. This is the field of cells. So where all of the molecules are initially, and then the red are individual molecules that are moving after activation, after they've been converted. And so what he did from these trajectories is he calculated the diffusion coefficient, and he got a very broad distribution, right? And so most of the polymerases are diffusing quickly, but there's a long tail where there are some that are moving more slowly. But without anything to really compare it to, that this doesn't really mean anything, right? So so he next looked at LAC-I. So we have integrated a LAC-O operator into the genome, and so LAC-I will bind to the operator and relatively stably stay bound to the DNA. And so what he's tracking here is the protein bound to DNA. And so what I hope you can see here is that the LAC-I moves much more slowly than the vast majority of polymerases, but there is an overlap. And so this overlap could be, you know, transcribing RNA polymerases that are bound to their template, right? And so what he did next was look at the density of tracks over time, and then calculate the mobility of the of each trajectory. And what he saw was that where there were high density of tracks, there was a low mobility. And so LAC-I, almost all of the proteins are bound, and they all move very slowly. But in the polymerase, you can see that there's high mobility states of polymerases moving throughout the nucleoid, but the slow ones are in these high density clusters, which have.
Sorry to interrupt. About 5 more minutes. Sorry to interrupt.
Okay. All right, no worries. This should be fine. And so what he did was he took then he set a threshold for density and found these clusters, and he assigned tracks that were inside the clusters. And so these are the red tracks that I've highlighted here, and these trajectories are slower than the majority of polymerases, but faster than LAC-I. And so now if we sort of dissect out that big distribution a little bit more, what you can see here, so LAC-I is the DNA bound. The green are trajectories that are found inside a droplet. The out are trajectories that are found outside these high density clusters. And then the in and out are trajectories that span the boundary, right? And so what you can see is in the in fraction, you actually see a bimodal distribution. And so slow molecules overlap with LAC-I. So we're hypothesizing that these are bound to the DNA and actively transcribing. But there's another fraction of molecules that are inside these dense clusters that move much more quickly than just the DNA. And so we're hypothesizing that these are molecules that are moving inside this liquid droplet, but that are not associated with the DNA. And then you also have molecules that are diffusing either in the cytoplasm or non-specifically hopping around the nucleoid. Okay, so that's sort of where we are. And now I'm gonna just take a minute or two to speculate. And so as I mentioned a little bit before, but I think the real elephant in the room in this field is, well, so what? You know, it's very clear that we can see clusters or droplets or granules or hubs or whatever condensates you want to call them assemble in cells, but so far it's been really hard to assign a very specific function to the process of phase separation, and not just, okay, it's a high concentration, so things happen there. And so the question is, does this have a function? And so I have no data on this. So this is kind of where we're at. But the working model that we have is that these clusters are maybe maintaining a high local concentration of polymerase such that it accelerates the reinitiation rate of transcription. And so when one polymerase finishes transcribing ribosomal RNA, rather than dissociating and diffusing throughout the cell, it will be able to rebind relatively quickly because it's still going to stay in this local droplet. But we have a lot of work to show this. Okay, so with that, I just want to say, so we think that that these RNAP clusters are phase separated. They assemble rapidly. They're reversible. They're sensitive to hexane diol. I didn't show you, but we also have some molecular data on disordered proteins that also seem to be required and have the same mobility states. And so the questions that we're interested in now that we can image this live is not just with single molecules, but whole cluster is to look at the kinetics of clustering and compare those with expectations from coarsening and Oswald ripening models that we've looked at for the nucleolus. How does it affect transcription rates? And more long term, can we manipulate the clustering and see changes in cell growth rate, cell size, and either move along this growth law, or can we move off of this growth law and break this law? And because if clustering allows this law to happen, then we should be able to perturb this clustering and sort of move perpendicular to this law. So with that, I just want to say that I was not the only one who had this brilliant idea. There are many examples coming out now of other possible phase separation processes in bacteria. So the RNase E forms droplets that may be important for RNA degradation. PopZ forms a condensate in the pole of Caulobacter cells, which is important for the asymmetric division. Poly P granules also seem to maybe phase separate and ripen in Pseudomonas. And there was a recent paper looking at partition complexes in E. coli. And I think the state of the field is now like, every time you see a bright spot in a cell, oh, it must be a droplet. And so what we've really been trying to do is come up with like rigorous quantitative tools to really test whether that is the case or not. And so with that, I just want to thank my lab. So this project really started with Anne-Marie, and Baljat really spearheaded the single molecule tracking. And James and Graydon were also involved in this work, but I didn't talk about their stuff. And I'd be happy to take questions, and thanks.
All right. That was fantastic stuff.
Very fun.
Yeah. So since you were in speculation mode, like Kat, yes, one of our alum, has a question. You know, what do you imagine is driving the switch between the clustered and the non-clustered phenotypes?
Yeah. So that's a great question. I have no idea. So there's a couple possibilities. So during the transition from stationary phase to log phase, there's a big change in gene expression. And so the level of, or the copy number of not only the polymerase itself, but also several transcription factors, and specifically NUS-A and NUS-B and NUS-E and NUS-G, and these are anti-termination factors, but they have this disordered domains that have been implicated in phase separation in other systems, and we've purified them, and in vitro they phase separate, and if you do various mutants in the cell, then you can perturb clustering. So that is one hypothesis. We haven't done a concentration curve to see if that is it, but so it could be protein concentration. It also could be some metabolite that changes. There's in the Caulobacter system, there's phosphorylation, like kinase cycles that are required. So yeah, we've just started dissecting the molecular mechanism, but there's a lot of candidates. Yeah.
So there are tons of really interesting questions, and maybe we'll do it in rapid fire format if you want to try it. There's so many questions. Maybe just keep short answers if you can. Of course, do whatever you want.
Sure.
The one was really I like this one a lot, which was I wanted to come back to the DNA binding hypothesis and how you've been able to reject it, or maybe if I'm in a more Bayesian mindset, like if you were to give relative weightings to the different proposals, in what way does the evidence favor the two? So yeah, maybe just can comment on where the two ideas are.
So I guess the other thing I should have emphasized is that they're not mutually exclusive. Well, in the sense that you can have some of the polymerases being bound to the DNA and phase separated condensate around it. And so what I would say that if it were purely DNA binding and no phase separation, I don't know why you would have molecules moving faster in that local space. You should get either the slow LAC-I mobility or the really fast, non-specifically bound mobility. But the fact that we see this intermediate population that is locally still in that cluster, that to me is pretty convincing.
Okay, so actually,
but if you want to,
no, I'm the messenger at the moment. So, okay. At least when he was in our course, Yaya was in Montreal, I think, if I remember correctly. He asks a good question, which is actually related to your point just now, which was if you make a construct in which you actually take, for example, an rRNA locus and you move it elsewhere, then the thought was, that you ought to actually gain a new spot. And actually, people for sure have mapped the position of sites on the chromosome in the cells. So you might actually have a coordinate system to be able to identify where it might go. So have a crack at that one.
Yeah, no, that's a great question. So people have actually done this. But way back before phase separation was a hypothesis. So there are 7 RNA operons in the genome, and you can delete 6 of them. Cells grow more slowly, but they're still fine. And so you get clustering in. You still see clustering, but the clusters are smaller. And people have also deleted all 7 and then put RNA or RNs on a plasmid, and there you get big clusters in the poles, and that's just where high copy number plasmids tend to hang out. So it's definitely moves with the DNA, but we haven't systematically looked at we haven't looked at the DNA. But there was another paper from Zhe relatively recently that did DNA fish and RNA polymerase and saw clusters co-localizing with the RRN genes.
So Fred Chang has a quick question, which I think is interesting and occurred to me as well, which was, he says, do you think that mRNA and or other RNAs are in the condensate?
Oh.
So, that's actually kind of very much related to Amy's talk yesterday, in the
the
subgenomic RNAs do not go in, and so that it's really a question about specificity. So anything you feel saying about that would be fun.
No, that's a fantastic question. We've just been on the working hypothesis assumption that it's mostly rRNA, only because of the relative abundance. It's like 90% of the transcription in this fast growth regime is rRNA, but we haven't looked or really considered that, but it's a fantastic question, and I just don't have much to go on it.
Okay. So it looks like Navish wants to ask something, which I think is kind of on this topic, and it brings you back to your thesis work. He says, does the cellular ATP concentration depend on growth media? Isn't there evidence that high ATP suppresses phase separation? This would contradict phase separation in rich media. So I figured that ATP has been on your mind in the past, so maybe you'll be able to quickly react to that one.
Yeah. No, that's awesome. So we haven't actually done, now that you say this, I'm embarrassed. We haven't actually done the ATP depletion experiment yet. It's certainly on the list. And I mean, the other, I guess I could go on about ATP for days, but we haven't done that in this context yet. So I don't really know yet.
It'll be fun to hear. I don't know what you would call a current student of Julie's as to whether that's a quote unquote lab mate of yours or not. But at any rate, Ricky has a question, which is she wants to know whether the effects of osmotic shock have any bearing on the clustering.
We have not tested that directly. It certainly has an effect on the chromosome sort of conformation. So I would be surprised if it didn't. But we haven't tested it directly. But it's an easy experiment and could and should maybe be done. Yeah.
Okay. Manuel has an order of magnitude question for you. It was really fun to see your order of magnitude estimates. So he's talking about the function of the condensates. At fast growth rates, RNAP is probably in the order of 10 to the 4th RNAP per cell. I agree with that. Comparing the loading rate of polymerases and the time to capture by pure diffusion without LLPS, do you actually need these clusters to accelerate transcription? So I love that. And he says, seems like there might be a simple order of magnitude estimate to be done there. And, I'll make a proposal, which is that he and I can meet and do it together if you want. But maybe you have some thoughts about it.
You don't need it.
Okay, cool. It'd be really fun to see that estimate. I'd love to.
Yeah. I don't remember the number off the top of my head. I have gone through that, and you don't, which is why I haven't emphasized it, because that kind of ruins the motivation for the whole project. But yeah, I think that I still don't know what the function of these are, right? Like, we see the clustering, it depends on growth rate, but yeah, I don't know.
But do we know that polymerase is getting to the locus at the diffusion limiter rate? That's the one. So I agree with the estimate that Manuel is proposing, but we know that the search mechanisms of transcription factors on the DNA are different than through 3D diffusion, right? So I don't know. Do we have that information? So just to refine it a little bit.
Yeah, with facilitated diffusion. Yeah, the truth is, like the question at the beginning of the talk, right? Like, if it's only 1 micron and you can diffuse across the cell in a second, like it's not. It might be a little bit faster with these clusters, but it's not necessary. So I don't know.
Great. So one question that came to my mind when you were showing the diffusion measurements, and I know you know about this better than I do, is what's happened. When you guys measure diffusion, was it actual diffusion? Is the scaling the right? Is the scaling the right number? The exponent the right number?
No. Well, we don't know, to be honest. So for the single molecule tracking, it's the trajectories aren't really long enough to get a really robust scaling. And so we just calculated an apparent diffusion coefficient, but it's almost certainly subdiffusive, but we haven't gotten a really good measure of that yet. But it's a great question, and we're trying to sort of optimize activation and light exposure stuff to try to get longer tracks. Yeah.
And I missed this. Did you show whether the clusters merge?
No.
Ah, okay. All right. Okay. So they seem to stay, but they seem to stay localized to the.
Yeah, on the order of like
10
minutes at 37 at least, they're pretty stable.
Okay.
But we haven't. Now we're starting to do the assembly disassembly with temperature and with growth media switches, and so that I really want to see. If you start and just see nucleation, presumably you would get nucleation multiple places, and then that would be a ripe time for. Sorry for ripe, I didn't mean pun. For fusion, yeah.
All right, so let's see. So there's a lot of questions. So we're gonna have to send you some of these.
Yeah, feel free to email me. I'm happy to follow up later.
What we'll do is and apologies to everybody. There are some of the questions, there's a bit of overlap, and we tried to ask the questions in our own words. So for example, Dong Yang, who's at UCSD, asked a similar question, one that I asked to Dong Yang, not trying to ignore you. I think maybe it's maybe a good time for us to close up, and thank you again for a great talk. It was really a lot of fun. And so let me just say, so tomorrow we have Alexander Awela, who is at the EMBL in Heidelberg, and an amazing guy, a really great speaker and a deep thinker, somebody I just love interacting with. He sent us a couple of students. And
Takahito's here today.
Yeah, he's here. And actually, some of you might remember, those of you that have been in the course, you remember we have an in-field and an out-of-field order of magnitude proposal. And I have to say that maybe Takahito's is the greatest of all time, because he tried to figure out if it's actually true if you could cry me a river. So anyway, he's Alexander's student. So Alexander is definitely somebody who works on physical biology writ large, and is going to talk to us probably about oscillations and multicellular organisms. But I actually didn't look at what his title is. But anyway, it should be fun. And I feel really good about the seminar series, that it's a nice way for us to spend an hour and 15 minutes every morning over these 3 weeks. And I really am grateful to the speakers for doing such a wonderful job of conveying their science, their enthusiasm for their science, their care, and also their willingness to not give the stump speech and show 60 data slides and all that stuff. So thanks again, Steph, and sending best wishes to all of you. And there's so many of you that I see on the participant list. I know that that Ernan shares this with me. It's just it's really fun to see all of you. I wish that your cameras were on, and I don't know, maybe I'll keep randomly calling out people in the coming days.
And Steph, we'll send you the questions, the
great.
remaining questions, in case there's something interesting, or, in case you want to follow up with people.
Sure thing.
All right.
All right, Ben Larson. I'll see you now. Take care.
Bye, everybody.